Thank you, Rusty, for that introduction, and I want to thank the Kavli Institute for supporting this event today. As it's already been said, this is a very important and new direction for neuro and for the microbiome and gut biology. And I'm excited to be a part of it today. So today I was going to tell you about, we do a lot of work on the microbiome and how the microbiome manipulates the gut-brain axis. But then I was thinking this morning how when I was in graduate school 10 years ago, all people cared about was host-pathogen interactions, and we completely ignored the microbiome. And now that we acknowledge the microbiome exists and we're seeing all the interesting things that we can learn from it, I think people have completely forgotten that pathogens can also be excellent tools to help us understand how our biology is working. And so I'm going to tell you a recent story that we've published about how host pathogens can manipulate the gut-brain axis. So in my lab, we're interested in really the general question of what does it mean to be healthy? And we want to understand the answer to that question so that we can ultimately learn how we can override physiological decline and how we can treat diseases. And the traditional approach to addressing that goal would be you take the body apart and you dissect particular pathway components or cells to understand how dysfunction of these pathways and cells can lead to disease. And we're taking a different approach in my lab. We're essentially putting the body back together because we want to understand how all of our physiological systems interact with each other so that we can ultimately manipulate these complex interactions to treat disease. And of course, the way we're going about addressing our goal is we're leveraging one of the, all the solutions that have been defined by evolution and leveraging one of the strongest drivers of biological complexity and innovation, and that's of course host microbe interactions, both beneficial interactions as well as pathogenic interactions. And I know when we think about pathogens, of course, pathogens are specialized microbes that have mechanisms to cause disease in us, but they, in theory, could also have evolved ways to promote health and basically have a balancing act of virulence in our bodies. And so it's important to think about these relationships from an evolutionary perspective because then our approach to using these microbes to treat disease and to promote health can actually make sense. So when we think about host-pathogen coevolution, both the host and the pathogen are adapting to not only maintain reproductive fitness, but they are also adapting to survive. And from the host perspective, that involves the selection of traits that minimize disease and promote health. And from the microbial perspective, that involves the selection of traits that enable the pathogen to acquire nutrients within the host to meet metabolic demands, selection of traits that enable the pathogen to replicate within the host, as well as the selection of traits that enable that pathogen to transmit to new hosts. And such adaptations are traditionally thought to arise from antagonistic host microbe coevolution, where you have a pathogen that's present in a host population, and those negative selective pressures drive the selection for traits in the host population that will in turn place negative selective pressures on the microbial population. And these sorts of evolutionary dynamics leads to the oscillation of these traits within the host and pathogen populations, and this is often referred to as the Red Queen effect in evolutionary biology. And such antagonistic host microbe coevolution is probably best exemplified by our traditional view of the immune system, the primary function of which we say is to sense and kill pathogens through the execution of what we would call resistance mechanisms. So pathogens, specific products of the pathogen or behaviors of the pathogen have driven the selection for our immune system, both the innate and the adaptive immune system, and the immune system responds by executing resistance mechanisms which protect the host by killing the pathogen. Now, a consequence of both resistance mechanisms and the pathogen are that pathology can also result. And pathology is not only going to have a negative impact on host fitness and survival, but it's also going to have a negative impact on pathogen fitness and survival. And because of that, I've been saying for about 10 years now that there has to be some other kind of evolution, host microbe evolution And I call this cooperative host microbe coevolution. And what I mean by this is that in addition to microbes driving the selection of antagonistic traits in the host population that protect the host by killing the microbe, that in theory, microbes that are present in host populations can drive the selection for traits that protect the host and promote health by targeting the physiology that's affected by that microbial interaction. Those traits are going to prevent physiological damage from even happening during this interaction. And both these are going to have very different evolutionary dynamics. They're going to go to fixation in the host population because they're not only promoting host fitness and survival, but also they have the potential to promote pathogen or microbe fitness and survival. Because the microbe has no reason to evolve an antagonistic trait against these such traits. And in support of this hypothesis or this proposal, when I was a graduate student, I had discovered that in addition to the immune system and microbial killing mechanisms, that animals also encode what we call a disease tolerance defense system. So these are traits that are absolutely essential for a host to survive microbial interactions, and they work by alleviating pathology that occurs during these interactions. So they're traits that promote host health and fitness while having a neutral to positive impact on microbial fitness and survival. Now, a logical extension of this discovery and our hypothesis is that microbes themselves could have evolved ways to induce this tolerance defense system in their hosts. And so one of the ways we're using the microbiome is we want to use this microbial community to teach us what these tolerance mechanisms are in the host and how we can effectively induce them in the host, and we've published some work on this. But today I'm going to tell you about our story where we ask the question of if pathogens can also induce tolerance defenses in their host and block pathology. And we explored this idea in the context of something called sickness-induced behaviors, specifically sickness-induced anorexia. So this is one of the stereotypical sickness behaviors that occurs in a variety of infectious diseases, along with changes in sleep patterns, fever, social withdrawal, lack of grooming happens. And the function of sickness-induced anorexia, it's not well understood. It has been proposed by myself and others to be an evolved behavioral strategy that promotes host defense during infection, but any beneficial function that sickness-induced anorexia has been demonstrated to have has been done in artificial host pathogen and infection systems. So we wanted to explore this idea using the mouse Salmonella Typhimurium system. We wanted to use this system because it is a naturally occurring host pathogen system. They have co-evolved together, and this is an orally acquired pathogen. It can cause both an intestinal disease as well as a systemic disease. In the intestinal phase, it can be spread to new hosts via the fecal-oral route, and it becomes systemic when it invades specific cells of the small intestine, and there it's picked up by innate immune cells, including macrophages and dendritic cells, and it can spread to various systemic organs, including the spleen and the liver. And we also wanted to use this system to test our hypothesis that pathogens can block pathology in their host because of this woman right here, Typhoid Mary. This was a cook in New York in the early 1900s, and she was asymptomatically persistently infected with a relative of Salmonella Typhimurium called Salmonella typhi. And unfortunately for the people she cooked for, she refused to wash her hands because she felt fine and she didn't see any reason for that. And as a result, she ended up infecting 50 plus people, and at least 2 people died from her food and the salmonella she flavored it with. So because she was asymptomatically persistently infected, it suggested to us that perhaps tolerance mechanisms could be at play in this relationship. So the first question we asked was, we took animals, we infected them orally with wild-type Salmonella, and we just asked the question of if we fast animals in a way that we can mimic a sickness-induced anorexic response, how does that affect Salmonella virulence? And what we found was that the animals that were fasted had increased virulence. The animals succumbed to the infection much quicker compared to the animals that were fed ad libitum during the infection. And what we found was that the levels of the pathogen were equivalent in the small intestine, in the fed animals and the fasted animals. But the fate of the pathogen was different depending on the feeding status of the host. So if we look at invasion of the pathogen in the fasted animals, we find that there's the percentage of hosts that have invasion events was significantly higher compared to the animals that were allowed to feed during the infection. But this when we looked at the transmission of the pathogen, we found the opposite. So the reduced invasion in the fed state was associated with increased transmission potential to new hosts, and the increased invasion in the fasted animals came at a cost to the transmission of the pathogen. Now, we were already studying a Salmonella effector protein called Salmonella leucine-rich repeat protein, or SLRP, and we were interested in this gene because if we infected animals with a mutant strain of this Salmonella, we found that it had increased virulence compared to animals orally infected with the parental strain, but the levels of the pathogen, the SLRP strain or the wild-type strain, they were equivalent. So there was increased virulence in the SLRP strain despite having equivalent levels of the pathogen in the small intestine. SLRP is expressed in the gut, so this suggested to us that SLRP could be acting at the level of the gut to promote health or regulate virulence in its host. What we found, what was interesting to us, was that if we force-fed the animals that were infected with the Delta SLRP strain, we can phenocopy the attenuated virulence in the animals that were orally infected with the parental strain. So we hypothesized that perhaps SLRP was modulating the feeding behavior and the anorexic response of the host during the infection. And that's exactly what we found to be the case. So this is just looking at one specific time point, but we see this over multiple time points over the course of the infection, that the animals infected with the Delta SLRP strain consume less food during the infection. They have a more severe anorexic response. And if we look at gene expression in the hypothalamus, which is the appetite control center of the brain, we find that there's differences in dysregulated expression of feeding programs in the hypothalamus. Consistent with our diet-restricted or fasting studies, when we looked at dissemination or invasion to systemic tissues, we found that the animals that were orally infected with the SLRP strain, these guys have a more severe anorexic response. They had an increased incident of invasion to systemic tissues, similar to the fasted wild-type infected animals. But if we force-fed the Delta SLRP strain, we could prevent the increased invasion to systemic sites. And again, this was negatively correlated with the transmission. So the increased invasion came at a cost of transmission in the Delta SLRP strain, and we could increase transmission by force-feeding the SLRP strain. And this is just showing survival if we orally infect wild-type if we orally infect animals with wild-type Salmonella, the primary host, or if we orally infect animals with SLRP, and then if we co-house them with a naive, uninfected animal, we find that consistent with the increased transmission, the wild-type animals that were co-housed with the primary infected wild-type mice, they have increased death kinetics. So there's more transmission to naive hosts, and they're dying faster in the secondary hosts. Now, we've done very careful analysis. I'm not going to show you all the data, but we've done very careful analysis of the kinetics of the anorexic response, invasion, as well as shedding, and that, coupled with our food restriction and force feeding models, allows us to conclude that the anorexic response is what's causing the invasion and the decreased dissemination. So what I've showed you so far is that fasting in the Salmonella model reduces host survival and transmission, and it increases virulence and invasion. The force feeding increases host survival and transmission, and that decreases invasion. We've identified a pathogen effector that blocks the anorexic response and regulates gene expression of feeding networks in the hypothalamus. This prevents invasion and promotes transmission and host survival. And in a naturally occurring host pathogen system, anorexia seems to be maladaptive for both the host as well as the pathogen, at least at the individual level. So of course we want to understand how all of this is working, and this is just my postdoc Sheila, who's sitting in the front. This is her story. She did an exhaustive list to eliminate the many different possibilities that could be how SLRP is modulating the anorexic response in the host. So we can talk about this in Q&A if you want more specifics. We considered a role for IL-1 beta because systemic IL-1 beta is believed to be the predominant mediator of sickness-induced anorexia. But when Sheila looked at levels of IL-1 beta in the serum and liver and spleen, she found no significant differences in IL-1 beta levels in the wild-type infected animals and the delta SLRP infected animals. What she found, though, what was really interesting to us, is that in the small intestine, in immune cells in the lamina propria, she found that there was increased levels of IL-1 beta as early as 24 hours post-infection in animals infected with the delta SLRP strain. So in the context of a salmonella infection, maturation of IL-1 beta involves the activation of a particular component of the innate immune system called the inflammasome, both the NLRC4 and NLRP3 inflammasome. These are multi-protein cytosolic complexes that are composed of an NLR sensor protein that senses a particular component of salmonella and other microbes. And also it involves caspase-1, and when there's activation of a particular microbial component, caspase-1 is cleaved, it's activated, and this leads to maturation of pro-inflammatory cytokines, including IL-1 beta and IL-18. And what Sheila found, of course, we thought perhaps the SLRP was inhibiting inflammasome activation, either directly or indirectly. So we looked at cleavage of caspase-1 in the wild-type and delta SLRP strain, and consistent with our hypothesis, we found that in wild-type animals there was less caspase-1 cleavage and activation compared to the animals that were infected with the delta SLRP strain. So SLRP seems to be inhibiting inflammasome activation, and that dampens IL-1 beta maturation in the small intestine of these animals. We, of course, wanted to test a role for the importance of IL-1 beta in the anorexic response, and so we administered recombinant IL-1 beta to animals that were infected with the wild-type Salmonella strain and looked at the anorexic response, and we found that we can induce anorexia in these animals. And we also looked at, in animals that were lacking IL-1 beta, how that affected the anorexic response in the context of a SLRP infection. And we found that if the animals lack IL-1 beta, we can prevent the anorexic response from happening in the context of a SLRP infection. We looked at the gene expression profile in the hypothalamus in these animals in the absence of IL-1 beta, and we found that the majority of genes that we pulled out from our RNA-seq analysis were the expression patterns were dependent on IL-1 beta. And of course, we wanted to ask, is IL-1 beta necessary for delta SLRP virulence? So this is looking at survival, and we see that if we infect IL-1 beta deficient animals with the delta SLRP strain, we can protect them from the virulence there. We see very little death in these animals. But what's important, I think the very important experiment, is Sheila then food restricted the delta SLRP infected IL-1 beta mice. And when she did that, she was able to phenocopy the virulence that she sees in Delta Slurpee infected wild-type animals. And this increased virulence is associated with increased invasion of the Delta Slurpee to systemic sites. And so that was a very important experiment because it allowed us to order the events and allowed us to conclude that IL-1 beta induces the anorexic response. It's the anorexic response that leads to the increased invasion and virulence and death in these animals. So how is the signal even getting to the brain to induce this response? We, of course, considered the hypothesis that the vagus nerve could be responsible for this. So we severed the vagus nerve to denervate the small intestine, and then challenged them with the Delta Slurpee strain. And we found when we did that, we can rescue the anorexic response as well as the IL-1 beta dependent gene expression pattern in the hypothalamus of these animals. And importantly, if just looking at feeding behavior in uninfected animals, I'm not showing you the data, but we find that vagotomy did not affect feeding behavior in an uninfected state, and this is looking at IL-1 beta levels in the small intestine, and those are not affected in the vagotomized animals. So by severing this nerve, we could block the signal to the hypothalamus to induce the anorexic response. So that allowed us to generate this model that we have here. So in the Slurpee deficient strain, we have the salmonella in the intestine. This, we get inflammasome activation and IL-1 beta production that signals to the hypothalamus via the vagus nerve to induce an anorexic program, and that leads to increased invasion of salmonella to systemic sites and increased salmonella virulence, and of course this comes at a cost to salmonella transmission and host survival. In the Slurpee state, and salmonella infections that have Slurpee, we can inhibit inflammasome activation in the small intestine. This minimizes IL-1 beta maturation, prevents this signal from getting to the hypothalamus. This allows food intake to occur. Salmonella transmits at higher levels associated with increased host survival and reduced invasion of the pathogen to systemic tissues. So I'm going to stop there, and I'm going to thank all the members of my lab, but specifically Sheila, who's right here. That she was the first author of that story. Alex and Danny also helped out with that story as well, and I think Sheila has a poster on this, so if you want to talk to her more about that, she would love that. I want to thank all my metabolism collaborators at Salk, including Ron and Ruben, and also Matt LeBlanc helped with pathology analyses for that story, and all of you for your attention. I'm happy to take questions. Thank you.
I'd like to point out for the rest of the speakers, that was a 25-minute talk, leaving great time for talks. Thank you. Open for questions. We have some.
Sure.
Yeah, sure.
Why does Salmonella make that peptide?
Well, so we have evidence. I mean, it promotes transmission. So if it doesn't have it, it comes at a fitness cost to salmonella, right? So Slurpee is one of the effectors that had no known function. We are the first to provide a function for it, and I like it because we, I think, provide an explanation for why it would even have it. If it doesn't, there's a fitness cost and survival cost.
Non-infective strains don't make Slurpee?
Yeah, so non-infective strains don't make Slurpee. Not so far, my analyses agree with that statement, yes. But I'd love to get the strain from Typhoid Mary and see, but I'm not sure that's going to be possible.
Hey,
Sarkis.
Janelle.
Hey.
Wonderful talk and beautiful story. But perhaps I missed something. In terms of anorexia, so anorexia is a behavior. So perhaps you can tell me how starvation models this behavior. I understand the force feeding experiment. I think that helps overcome the issue of any technical issue or any technical variables. But again, was there a baseline anorexia in the infected animals, and did those animals have many of those same pathways that you showed us?
So in a wild-type infected salmonella infection, there will be some anorexia that occurs. So there's already a baseline, and there's increased levels in the context of it's more dramatic with the SLRP. For our fasting experiments, we didn't do starvation because I agree starvation is not the right thing to do. So what Sheila did was we knew how much the SLRP-infected animals were consuming, and so she basically did pairwise feeding. So she matched the, what we're calling fasted or food-restricted wild-type infected animals, gave them the exact same amount of food that we saw for that time period in the SLRP infection. And she gave it during multiple times of the day, so we try to mimic it as much as we could with that model.
And in that baseline anorexic state, if you force feed, can you overcome whatever phenotypes you're observing?
If we force feed a wild type infected salmonella, that's also beneficial. Yeah.
Terrific.
And my mother had typhoid when I was 2 years of age. And she would carry it because we found bacteria in the gallbladder. And stones were removed 15 years later. Is there a difference among some people who develop the carrier state?
So I couldn't hear most of what you said. Someone had salmonella at a young age?
When I was 2 years of age,
Uh-huh.
she had typhoid fever.
Okay.
And then when she was operated on the gallbladder stones, 15 years later, the bacteria was found in the gallbladder stones.
Uh-huh.
The question is, do some people have a propensity for developing such a carrier state?
So they knew it was salmonella in the gallbladder?
Yes.
Yeah. So I guess, well, yeah, so typhoid Mary is an example of a persistent carrier state, right? So I guess the question would be, is it the same infection that she had when she was younger, or is it a new infection that happened? Don't know. But yes, it's one of the reasons why we use this model, because there are carrier states.
So what's upstream of IL-1? What is activating the elevated levels of IL-1?
So that's the inflammasome. So NLR proteins with caspase-1, they recognize various components of salmonella, and that leads to cleavage of caspase-1 and
So
activation.
it's not NF-kappa B mediated,
Well, NF-kappa B you need for transcription of.
but its machinery is not changing, it's just there.
So Sheila looked at transcription levels, and that was not effective. So it was definitely the maturation of it that was affected. Now, whether SLRP is directly inhibiting the inflammasome or indirectly, we don't know that. There's a SLRP homologue in Yersinia called YOPJ that's been shown to directly inhibit the inflammasome activation, IL-1 beta maturation. And their model, there's also an NLS sequence in YOPJ and SLRP. So their model is that YOPJ binds to caspase-1 and it sequesters caspase-1 in the nucleus, preventing inflammasome formation from even happening.
If typhoid Mary analogy were to happen now, what would be your recommendation? Well, no.
Wash your hands.
No.
Don't cook for people.
That sort of thing, but I mean, the targeting mechanism, would you be upstream? Would you? go to
Oh, well, the
IL-1?
typhoid Marys are fine, right? So
Well, okay, let's say the infection, would you recommend blocking IL-1 beta, or would you go further downstream?
So it depends on this is even affected in, I think yeah, I mean, would you even want to target somebody that is perfectly
Healthy?
fine with it?
Yeah,
I don't know, quarantiner.
yeah.
Yeah.
Okay,
Okay,
thank you.
thank
Janelle.
you.
Really.